Hello everyone, hopefully you can see me clearly now. Hopefully I'm all coming through and the sound doesn't sound too terrible. Welcome to our virtual classroom here at Virtual Selwyn College. We're going to be doing some virtual online learning. I'm hoping that everything is coming through clearly. Could I just get you to say in the chat if all the sound is coming through, if you can hear me, if you can see everything, that would be good to know. It might be a tad little glitchy. My little baby computer is trying its darndest to try and keep up with showing everything. The good thing is this will all be recorded for you, and the recordings tend to be a little bit more high fidelity after they've gone through some processing. So if you miss anything, or if you just want to review the content that we go over today, you're welcome to have a little bit of a view of the video after the fact. Okay, what are we going to be doing today? Well, good question. Our content for today is looking at meiosis or meiosis, looking at genes and alleles, and looking at genotype and phenotype. Now you will already know some of this for the content that we've been teaching so far. So for example, you may immediately be able to identify that those images over, let me see if I point in the right direction. I think it's over this way. Yep, got it. Over this way is of 2 chromosomes there with 2 different genes. For those of you who know your genetics history, you might recognize these as being part of Mendel's Ps, and we'll talk about Mendel in a later week, one of the OGs when it comes to studying genetics. You'll notice that there's 2 traits identified on there. There is the height gene and the P gene, or really like what the phenotype of the P will look like. And the particular organism that we can see has 2 tall alleles. We're remembering alleles are different forms of the same gene, so presumably there might be a short allele or maybe even a medium-sized allele. And we've got our P genes there. We've got our smooth P gene and our wrinkled P gene. Now, if we knew what these genetics of this plant were, and we found what the P's actually look like, and the P's looked smooth, that might tell us a little something about whether smooth or wrinkled are dominant. What it would tell us is that the smooth is the dominant one. If we had smooth offspring given that we have this big letter, small letter, hopefully we're remembering what that's called, because it's out of Mr. Wills's mind, which is not ideal, but don't worry, we'll be going through that in a later week, and I'll make sure that I remember it from then. Just going to check our live stream, making sure it's all good. Okay, let's dive into the content for this week. I don't want to keep you for too long, and let's dive into it. So these slides will all be up for you on Google Classroom, along with an assignment that the Year 11 coordinator, Miss Kumar, has put together. Your first task was to go through these terms. Now, hopefully, we went through these last week. Hopefully, you can see the term that Mr. Wills was just forgetting. That's right. It was heterozygous, right? Hetero meaning different, homo meaning the same, zygous referring to the zygote. So heterozygous would have been this example. If we flick back to that, you can see wrinkled and smooth, 2 different genes, 2 different alleles, 1 big letter, 1 little letter. That would have given us our heterozygous for that particular gene. We've also got homozygous, referring to 2 of the same. Now we've got genotype and phenotype. Hope you remember it. Genotype is the type of genes you have. Phenotype is what your physical traits are. So being if it was a big W and a little w, for example, for the wrinkled genes or the smooth genes, so those would be the genotype of that organism, what combination of those they had. The phenotype would have been whether the P came out as smooth or wrinkled. We've got some other terms here which hopefully we remember and are able to write in. That's your first little task. What I'd encourage you to do is, if you started on those cue cards, when we were doing that work last week or the week before, I'd like you to add in those if you're missing any, but hopefully you should have all of those. Ms. Kumar's put in a little series of the tirotaki for this lesson, so the lesson plan. We're going through some notes on meiosis and those stages. There's a little task for you, and there's a video that will hopefully help you out. So we'll step through those, and hopefully this will assist your study in working through those. By the way, as we're going through, if you have any questions, you're welcome to put them in the chat. I'll be flicking back and forth, so hopefully I will see those. If you have any questions after the fact, you're welcome to either put them in the comments or bring them to our Google Meet, which will be tomorrow. Let me check the time. That'll be period 3, so at 11:35 a.m. tomorrow. All right, meiosis notes. So first thing about meiosis, it's different to mitosis. Mitosis occurs everywhere in your body except the gametes. Meiosis occurs for producing sex cells. Why? Well, sex cells only need half of the genetic material, right? They're haploid, right? Ha meaning, or HA, referring to one set, ploid referring to the chromosomes. Diploid, like most other cells in our body, di means 2, and ploid means chromosome. So 2 sets of chromosomes in all the rest of the cells in our body, and half of that, 1 set, haploid, in our gametes there. In meiosis, we take a cell that is diploid and we use it to produce a bunch of cells that are haploid. These processes are largely the same, whether we're making a male sex cell or a female sex cell. So we talk about meiosis in more general terms here. If you're interested in the specifics, there are a bunch of interesting differences that occur when you're producing either an egg or a sperm, but we'll talk about that at a later stage. The key thing to then remember is that these sex cells sit around for a while, but eventually one of those sex cells will go along and meet another sex cell, and they can then join together and create another diploid cell. In this case, we'd call that a zygote, and that goes on to become a new organism. Hopefully that's all clear so far. In this particular slide, you've got 2 tasks. You can complete either this EP set of questions on meiosis or the SciPad page, or you can complete both of them if you feel like you need that kind of extension. I would challenge you to pick the one that might be more challenging for you, as that will challenge you more and further develop your learning. There's also a little video here by the Amoeba Sisters, fantastic channel if you want to look at some good biology content. Yeah, so that's there for you to review. And again, there's these notes in here, another page in SciPad and another Education Perfect task related to this. What I'm going to do is quickly step through the stages of meiosis with you so that hopefully we understand it. If you've got any questions while we're working through this, you can post them in the chat and hopefully I will answer them promptly. Okay, in meiosis, we start off with the same type of cell that we do with mitosis, and that is a diploid cell with 2 sets of chromosomes. You can tell that here because if I move my cursor over, you can see we've got half the chromosomes that are paternally derived from the father, and half that are derived maternally from the mother. You can see in this cell here, it undergoes DNA replication. And we talked about that process last week, where the DNA unwinds and is replicated in a semi-conservative way, so that you have 2 copies, each of which contains half of the original strand. Once you've got pairs of duplicated chromosomes, these chromosomes line up in the center of the cell. Hopefully you can remember which stage that was from our discussions last week. If you can't, refer to your notes. When these line up, they line up on what is known as the spindle, which is the name we give for the middle of the cell, and they are pulled apart. So let's say this was chromosome 10 for us. both of our new cells after this first division will have one copy of chromosome 10. But that chromosome won't just be a single chromosome, it'll be sister chromatids. They'll bring over both copies of the paternally derived or both copies of the maternally derived. Now, I'll talk to you at the end about why there's a little bit of green on this blue one and why there's a little bit of blue on this green one. But let's step through it. That is our first instance of cell division. You see here, we're continuing over on this side. We now have two cells, where originally we had one, each of which has half of the original chromosomes. But remember, those chromosomes are still replicated. This is the same amount of DNA, it's just doubled up. So we go through a second process of cell division where these sister chromatids are pulled apart at the centromere, leaving us with two copies of the paternally derived chromosomes in two cells and two copies of the maternally derived chromosome in two different cells. That gives us a total of 4 cells at the end. Now you'll recall from mitosis, you were left with 2 cells at the end, containing both maternally and paternally derived copies of the chromosomes. Those are some of the key differences and steps if we step through it. So first thing we do is we replicate all the DNA. Second thing we do is line up in the center of the cell. Third thing we do is move one half of our sister chromatids, of our sets of sister chromatids onto one chromosome and our other half onto the other one. So for each chromosome, for each cell after cell division 1, those cells will contain either the maternal copy or the paternal copy, usually not both, right? In pretty much all cases, and you can think of this as pretty much a rule for NCEA Level 1. After that first cell division in meiosis, you have one copy of each chromosome. So each of these cells will have a copy of chromosome 10. They only show one chromosome here for simplicity's sake, but you can imagine in human gamete reproduction, these cells will contain either one copy of either the paternal copy of chromosome 10 or the maternal copy of chromosome 10. They'll also contain either the paternal copy of chromosome 15 or the maternal copy of chromosome 15. Those cells then have to undergo a second division where those sister chromatids are split, and we end up with this at the bottom here. So from one diploid cell, we have 4 haploid cells. Compare that to mitosis, where we have one diploid cell producing 2 diploid cells. Hopefully that all makes sense so far. I'll show you this diagram. It'll be a little bit hard to see, but this is next door to mitotic cell division, right? Showing this example again, hopefully you can see that there are some key differences there. You've got one cell division occurring in mitosis, where you've got 2 occurring in meiosis. In meiosis and mitosis, you both have a doubling of all of the DNA, but during the course of mitosis, you have one diploid cell becoming 2 diploid cells, whereas in meiosis, you have one diploid cell becoming 4 haploid cells. Hopefully that all makes sense so far. Okay, we're going to move on to the next chunk of content, which is on genes and alleles. And don't worry if that's a little bit tough. I'd encourage you to complete those practice questions, either in Education Perfect or in the SciPad pages using the online SciPad. And if it's still a big challenge, save your questions for the Google Meet, and we'll go through them tomorrow. Okay, first little mahia. How do genes make proteins? Now, some of us might have some prior knowledge on this, and it's okay if we do. There is a process that we will be learning about both today and over the course of the rest of this term, about how the code for life, which is deoxyribonucleic acid, which we've talked about a little bit already this term, gets read and then the message is then sent to the machines that then produce these little proteins which do various jobs around the cell. Now, the key thing that we want you to take away from this is that in order for us to have any trait, whatever it may be, there are proteins involved. Whether you've got earlobes like I do, or whether you don't have earlobes, that is defined by the DNA sequence that you have and the protein that that sequence codes for. It's also true that those traits are influenced by the environment, but there's a lot of traits that aren't influenced by the environment. It's just the luck of the draw whether you get it. We've talked about some of those in class already. Eye color is a perfect example. There are a number of different genes that code for different parts of eye color. Each of those genes controls the creation or manufacturing process of making a protein or a group of proteins. And those proteins then work together to give us the color of our eyes. And those of us who know a little bit about evolutionary history, originally we all had brown eyes. But it wasn't until peoples migrated out of Africa and moved further north into climates where there were great deals of snow. This caused a number of changes. One of those changes was the mutation to produce blue eyes like mine. So any of you with blue eyes, you're a mutant, and that's okay. Just like me. That's good though, because in those highly snowy environments where you've got a lot of reflected sunlight coming off, blue eyes were advantageous as they allowed you a better ability to see and reduced chances of eyes being damaged, at least as far as I can recall. There's a whole bunch of other DNA to protein to traits that we'll learn about over the course of this term. There's a little video for you here where, again, the amazing channel, the Amoeba Sisters, go through the process of how alleles and genes work and how they code for various things. There's also, again, these SciPad pages, which I'd encourage you to take a look at. The key thing that we need you to know is that DNA, that sequence, is the code that produces that protein. I'm going to go into more detail in this in the weeks to come. But it's that code for life that codes for each of these little molecular machines that do jobs inside our cells and allow us to be able to survive and thrive. Now that brings us to the end of lesson 2. We're flying through this. Lesson 3, our last lesson for this week, is on genotype and phenotype. If you've done the previous tasks, then you're absolutely set. Our goal for today is to have a little bit of a kōrero about page 194 and 195 of your SciPad. Now I'm going to pop up page 194. There's some pre-text in here which I'd encourage you to read using the link that's on those slides. What we need to talk about here is essentially just like your average bog standard. So just make sure I sign in the people who've said hi in the chat. Hello to all of you. This is our sort of bog standard human reproduction, human production of a zygote that we're talking about here. So you see with males, by and large you have the code XY. Now we talked about exceptions to this, but we'll use this as our general rule of thumb. In humans, we have our sex chromosomes, our 26th set of chromosomes, we give the name sex chromosomes, and instead of giving them numbers, we give them either an X or a Y. Now why do we do this? Well, actually, if you look at them under a microscope after they've become sister chromatids, the X's look like this, and the Y's look much smaller, much smaller. And that relates to some interesting biology, which you're welcome to have a little bit of a Google of. But we can tell very clearly if an organism, if a zygote of a human is X or XY just by looking at the shape of those chromosomes. If you have somebody who is XX and they produce some gametes through the process of meiosis or meiosis, both of those original X chromosomes will duplicate, so you will then have two sets of sister chromatid X chromosomes. Those will then be split, as we remember from the stages of meiosis, in that first process of cell division. So you have half having one X, half having the other X. Those then are divided again as those sister chromatids are ripped apart, and you end up with 4 gametes which are haploid, containing only one X chromosome. However, for males, when producing sperm, the same process occurs, but one of those chromosomes is Y. So half of your DNA is, half of the X chromosomes, half of the sex chromosomes are X, so that X chromosome is split is replicated. So you have 2 X's and 2 Y's in that original cell that then divide. You then have your 2 copies of X and your 2 copies of Y in 2 cells after that first cell division of meiosis. Those cells then replicate again, or split again, and that replicated X chromosome is split into either 2 copies of X, so you've got 1 X over here, 1 X over here, or you have Y, 1 Y chromosome here, 1 Y chromosome there. Now, when those gametes meet a gamete, so for example, if you have a sperm that is X and it meets an ova or an egg that is also X, the resulting zygote will have 2 X chromosomes. If, however, you have a Y chromosome sperm meet with one of the ovas, both of which are X, then that creates the offspring XY. So, given that information, our possibilities we can look at here are X plus X is XX, X plus X is XX, X and Y is XY. Usually just by convention we write the Y afterwards because we like to be alphabetically ordered. And here we have the Y plus the X, which leads to XY. So given that these chances of getting each of these either sperm or eggs are equally likely, the probability that you get an XY offspring, which we typically classify as male, or biologically male, or XX, which we classify as biologically female. What are those probabilities? Well, there's a 50% chance that you get a Y chromosome and 50% chance that you get an X chromosome. Given those odds, that gives you a 50% chance of being born male or 50% chance of being born female. Remember that biological sex is different to like sociological gender. So what we're talking about here is biological sex or sex at birth. Remember, there are also other complications that come along with this too, but we need to understand the fundamentals first. If a couple already had 2 children, both of whom were girls, what's the probability that their next baby will be a boy? This is something that's known as the hot hand fallacy. If someone makes a bunch of shots in basketball, does that mean they're necessarily more likely to get the next shot in, or they're almost certain to get the next shot in? No, they've always got the same probability, 50%. Right? What's the probability that the next baby will be a boy? It's 50-50, whether or not they get the X or the Y, unless there's some other piece of information we haven't been told about. Say, for example, why sperm don't develop properly or have tails that don't work as effectively in this particular man. So then you have to write in your answer there. Hopefully that all makes sense. If anyone has any questions, now would be a great time to post them in the chat. Okay. While we're looking here, I'll pop up the next little SciPad page, and that's talking about different rats. So we've got rats here with genotypes. We use big B to signify that the gene is dominant, and little b to signify that it is recessive. We have 2 copies of the recessive gene. We get a white phenotype of fur. We have 2 copies, or a heterozygous organism. Here we get a black phenotype. So the key thing, the length of fur of the rat is also controlled by the gene. Long fur is big L, short fur is little l. which allele is dominant, hopefully we can figure out from our prior knowledge that we tend to use our capital letters to refer to dominant traits. So you can write those in there. Our recessive traits we tend to save for our lowercase letters. Describe the phenotype of individuals with the following genotype. Well, if it's big L, that's homozygous dominant, right? But the phenotype, so the outcome, will be long fur, won't it? Because they have 2 copies of the same allele there. For here, we've got 2 little L's, which is short fur, same thing there. Here we have a heterozygous individual, so they have 1 big L and 1 little L, right? Now we know that the dominant allele is always expressed ahead of the recessive allele if both are present. And we'll go into the mechanisms behind that in more detail in an upcoming lesson. But here, you've got a heterozygous individual. Same with this one here. It'll be the long fur, the dominant, which is expressed, so the phenotype is dominant. So the genotype and phenotype of the rat that is homozygous recessive. Here we go again. Homozygous recessive, that's 2 little L's. Heterozygous, 1 of each, big L, little L. Homozygous dominant, 2 big L's. Excellent. Explain why the rat has long fur, can have 2 possible genotypes. I'll leave that up to you to complete. Hopefully that's been enough information for you to get by on that one. Again, similar answer there for E. Hopefully we're all able to figure that out. We'll go through the mechanisms behind this in more detail in the weeks to come. The last little thing here is an extension task, and this is an optional task which you may want to do if you're feeling really in the mood for some more biology and science this week, like I am. There are some exam questions on No Brain Too Small, and what these will do is really test your mettle, and what I'd encourage you to do is if you finish some and you'd like me to have a little bit of a go at marking them, take a photograph or scan in your answer and email it to me or upload it to Google Classroom onto the assignment. If you do that, I'll go through and tell you what you're doing well and what you need to work on, and that will really prepare you well for that assessment at the end of the year. It'll also, I think, challenge just how well you truly understand these aspects. Now lastly, you would have seen at the bottom of the slides there was this, our lesson 4, so that's what we'll be doing tomorrow. Tomorrow we'll just have a little bit of a Q&A during our Google Meet, and we'll follow that up with a little bit of a Kahoot because I thought that might be fun. And that about does it for us today. That brings us to the end of everything that we needed to cover. If you've got any questions, you're welcome to post them in the chat now. I'll hang around for a minute or two in case anyone has something they want to ask. Otherwise, have a lovely rest of your day. Look after yourselves and your whānau. I hope you get the opportunity to go do some exercise, even though the weather's been pretty shocking recently. But otherwise, have a lovely rest of your day, and hopefully I'm looking forward to seeing you all in person soon. Okay, mā te wā.